This podcast is brought to you by Craft Beer and Brewing Magazine for those that love to make and drink great beer. To learn more or to subscribe, visit beerandbrewing.com or find us on social media at Craft Beer Brew. It's episode 413 of the Craft Beer and Brewing Podcast. This episode, we're going to focus on virus-free hops. Joining me are two experts in the subject, Megan Twomey. Welcome to the podcast, Megan.
Hi, Jamie. Thank you for having me.
Megan's with Latitude 46, and then, of course, Ryan Christian of Yakima Chief Ranches. Welcome to the podcast, Ryan.
Yeah, thanks, Jamie. It's a pleasure to be here.
Both of you all are experts in hop biology and the way that hops grow. The way that this episode came to be, couple weeks ago, Joe from Yakima Chief Ranches reached out. I guess he'd had some conversations with brewers, but I'd also hear on the podcast, especially in the context of New Zealand, we were talking about plant stresses. We were talking about the way that may, affect hop expression in New Zealand. And I know I made an offhand comment there about virus-free hops and the way that may, or that how anecdotally had been shared with me from brewers about how that may impact that kind of flavor and aroma expression. Joe thought there might be some information that went another direction, or at least that would expand the conversation around that and let us look at this subject of whether hop virus as a plant stressor might be impacting flavor and aroma in some sort of way. And so we got the conversation started. Again, Megan's been active in this. Ryan's been active in this. We thought we'd put a conversation here together to talk about everything from like a broad view. What are hops viruses? How are growers mitigating that, especially in the growing area of the Pacific Northwest? What technologies are hops growers using to maintain virus-free stocks, to breed and select for that kind of viral resistance? What current virus-free plantings look like? Agronomic performance, data around sensory and oil content and whatnot of those plants. We want to definitely control, when we look at this through this conversation for all the other variables, because hops are this giant multivariate equation: terroir, growing season, other kinds of variations in inputs, plant age. All of these things affect the way that hops themselves express beyond just simply by the virus-free nature of those plants or how they're bred or whatnot. And so we want to kind of look at that from a big picture, but as well as a mechanical kind of standpoint. And we're going to get into the weeds on this one. But first, for years, G&D Chillers has chilled the beers you love, partnering with 3,000 plus breweries across North America and beyond. They're proud of the cool partnerships they've built over the past 30 years, because brewing doesn't stop at 5 o'clock, and nor do they. They use quality components, expert craftsmanship, and constant innovation. With their 24/7 service and support, your brewery will never stop. Remote monitor your chiller for simple and fast access to all the information you need, and gain peace of mind your operation is running smoothly. And brewers, what if your yeast could do more than just ferment? With Berkeley Yeast, it can. They bioengineer ordinary strains and make them extraordinary, enhancing the flavors you want and eliminating the ones you don't. Now, in addition to competitive pricing, they're offering free overnight shipping on 5+ barrel pitches. Try it out. If you don't love it, they'll happily refund you. So why settle for ordinary when you can brew with better yeast? Visit berkeleyyeast.com to learn more and start brewing with science on your side. And as breweries expand beyond beer into other segments like mocktails and CBD beverages, Old Orchard is here to help. They can help formulate custom blends featuring specialty ingredients. Whether trending flavor additions or nostalgic favorites, the next best thing is around the corner at Old Orchard. More information and free samples are waiting at oldorchard.com/brewer. That's oldorchard.com/brewer. Megan, let's start with you. Give us a little bit of background, how you went down this path, through plant biology, ended up where you are, and then tell us a little bit about Latitude 46, its relationship with Roy Farms, Greenacre, and others, and how all of that works. Give us that background story.
Sure. Well, I did my graduate work in plant pollination proteins at Western Washington University. And I kind of took a summer job managing a research greenhouse for one of the merchants in town in hops, and that was back in 2009. And so obviously it stuck, and I'm still around. So I was at the very first NCPN Hops meeting in 2010 when hops applied to actually be part of the national program. And so
that's the National Clean.
the National Clean Plant Center.
Network.
Yes. So the NCPN stands for National Clean Plant Center. Is the only certified clean planting material resource for many crops in the U.S. It entails all tree fruits, grapevine, rose, sweet potatoes, and hops. There might be a couple of other crops in there that I'm neglecting. Hops only has one facility that provides a service, and that's located at Washington State University in Kosser. So back in 2010, hops applied to be part of this the certified clean planting program, because hop stunt viroid was discovered in 2004. And so at this point, it was the first major deleterious virus, or viroid, we'll get into that in a little bit, really discovered in the hop industry to have pretty drastic effects on yield. And this includes both cone yield and alpha yield in various varieties. So that kind of instigated the industry for wanting to join in this program. And since then, so now that it's been around for about 15 years now, the program has served both private and public varieties. So you could get cultivars cleaned up there, whether they're proprietary or public, and you can either take them back if they're proprietary or they get released to the public. So currently the NCPN has 52 varieties that are publicly available, and that's anybody can order from them. So the resource is there to start clean planted material if you're interested in public varieties. And then I'm sure Ryan could talk a little bit more on the proprietary cleanup and alternatives to the NCPN. But for right now, that's the only public resource available for growers and brewers alike to source virus-free material. So after I started in the industry in 2009, I went to Oregon State University and was the lead tech in the plant pathology program there for several years before coming to manage the Latitude 46 breeding program. We started as the American Dwarf Hop Association, which focused primarily on low trellis, high alpha breeding. But since then, we've expanded with various germplasm collections into breeding for standard trellis height and low trellis, and for a wide variety of traits. So right now, Latitude 46 is our latest iteration, and we are co-owned by Green Acre Farms and Roy Farms. And our main varieties in our program are Azaka, Pico, Summit, Adina, and Urillo.
And then some experimentals. I remember I was looking at my phone yesterday, and I have a note on there, Latitude 46, 1631, that dated back to CBC last year. Someone was rubbing it. They told me about it. I can't remember who it was, but I made a note just so I wouldn't forget that. And here you are again. Now we're talking to you here from Latitude 46. And at some point here, I'm going to learn more about that hop.
Yes.
I definitely want to talk more about cleaning up and what that entails, because that sounds interesting. But before we do that, Ryan, why don't you give us some background on what you do and your kind of career path here?
Yeah. So, I mean, I've been interested in plants my whole life. I was that kid that was really interested in getting seed catalogs in the mail and just geek out over that every year, what to plant in the garden and everything. And as I went to college, I studied agricultural biotechnology. Got kind of interested in the more genetics oriented route. Got connected with the lab there that ended up doing my PhD with. And during my PhD, got into homebrewing. I didn't have any connections with the hop industry at that point at all. But I was really interested in craft brewing. Probably didn't brew anything all that exciting at the time, but at least got interested in the plant and what it could do. In grad school, I was studying primarily chloroplast evolutionary biology, so kind of understanding how it differed from species to species. And there's interesting tie-ins there with hops, that there are unique biochemical pathways to hops, like there's unique biochemical pathways to hops, including the alpha-beta pathway that are found in no other species. So it was things like that I was studying primarily in my for my dissertation work, my PhD. And right in the last couple months, as I was finishing up, I got connected with the hop industry and found that Yakima Chief Ranches was looking for a technical director at the time, and I reached out to Jason Peralt, and he thought that I had the stuff needed for the job. So, and one thing led to another and ended up here. So my role here is primarily on the breeding and genetics side, as well as in the plant quality side for maintaining virus-free hops for our proprietary varieties. So my kind of expertise here is anywhere from the initial crossing work all the way through management of the brands once they're released.
Cool. And we definitely will talk about why this matters to brewers, why we would have this kind of conversation, because everything flows downhill from, that field, everything from the cost that brewers pay, because efficiency and agronomic performance is incredibly important both to grower and to the ultimate price for that, but also the quality of aroma and flavor that is delivered through those consistently across the field also, because, the more variability that enters into any given kind of field, the less kind of consistency you might even find within, that field, which then reflects in the bales that people get and the pellets and everything else. And so maintaining plant health is incredibly important, even if it may seem like a definitely upstream kind of process for brewers. Anyway, we're going to talk about what exactly that means. But first, both classic and contemporary indie hops Lorien was developed with crisp, low-gravity beer in mind, a nod to the past with an eye to the future. Lorien seamlessly combines traditional elements of European noble hops with an elegant twang of American modernity. Low alpha and squeaky clean, Lorien provides a refined, smooth bitterness to any lager and delivers enticing flavors and aromas of lemon and lime zest, fresh melon, wildflowers, sweet hay, and cinnamon spice. Refreshing the past for a present time, Lorien's classic profile and distinct finish are conducive to celebration and appetite and evoke feelings of a grassy meadow near a cold mountain stream. Summer is near and Lorien awaits. Learn more about Lorien and the rest of Indie's varieties at www.indiehops.com. Indie Hops, life is short. Let's make it flavorful. And are you a craft brewery looking to elevate your branding? Steel Chill Cups are the perfect promotional tool. Crafted from 100% recyclable steel and proudly made in the USA, these 16-ounce cups come in vibrant red, blue, green, and gray. Designed to keep beverages cold and frosty to the touch, they enhance the drinking experience at festivals, tastings, and tap rooms. Customizable branding options allow breweries to showcase logos and unique designs, boosting brand recognition. These stylish cups not only serve a practical purpose, but also become keepsakes for beer lovers. Discover how Steel Chill Cups can put your brand on every pour. Visit steelchillcups.com today. Megan, why don't you start off with a little bit of an overview of hops virus and what it is? And I guess we talked about virus, but you also mentioned viroid and are parsing a difference between those. Help explain to me, a general layperson, when it comes to the science and biology of hops, what we're talking about here, what these things are that you all are actively working to either clean up or protect against, breed more resistance towards, and whatnot, how those affect the plants and what that actually means.
Sure. So I guess just to get kind of a broad overview on the viral classes that we have present here in the U.S. The first class would be the carlaviruses. These are aphid-transmitted, aphid-vectored viruses. The biggest virus of economic concern in that group is going to be hop mosaic virus. And so the way that these viruses are typically controlled is by mitigating your aphid load through insecticidal controls early on in the season. And typically a farm practice will be to use something that persists for 3 to 4 months. So they only have to do a simple treatment for aphids if it's done early on, almost like in a prophylactic way. The second major class...
So a spray that'll happen early,
Mm-hmm.
so that, it will wear off by the time harvest comes around, and you have a nice kind of clean space there for, before harvest happens.
For sure. And historically, this has been an imidacloprid, which is becoming harder to use now due to restrictions for export crops into the EU. And so farmers are kind of having to change it up a bit. And there are some good biologics out there, but, you really want to get those aphids under control early. So, typically, like on our farm, we'll usually treat prophylactically most fields in like mid to late June just to carry us through till harvest. So that because aphids, aside from being viral transmitters, viral vectors, can also cause cone quality issues if they get into the cones through their honeydew, which tends to be colonized by different fungal species, leading to sooty mold and an overall very unpleasant organoleptic factors that along with this. So there are multiple reasons to control aphids, but again, like it is the main vector for for hop mosaic virus and the hop latent viruses. Now, the second class of viruses is the Eiler viruses. And the most important virus of note in this group is Prenus necrotic ring spot virus, also known as apple mosaic virus. So this virus tends to cause oak leaf necrotic regions on leaves and will cause stunting and decrease in yield and potential alpha acid effects over time. These do tend to be variety specific, as with all viruses and viros. They tend to have different effects on some varieties more than others. For whatever reason, the alpha varieties tend to tolerate these more than some of the more delicate aroma varieties. But again, like this, the consequences of a viral infection tend to be compounded with multiple infections. So just having one, a hop can tolerate fairly well. However, once you get these in a viral complex, the effects tend to compound. And then the last class isn't even technically a virus. It's our viroid class. What these are is small, around 294 nucleotide circular RNA species that basically insert themselves in the hop regulatory genes throughout the genome and hop and proliferate very rapidly. The Eiler viruses and the viroids are entirely mechanically transmitted. There is some evidence that viroids can be seed transmitted or at least be detected in seeds, but actual transmission from seed is yet to be proven. The biggest member in the viroid family that's of the most economic importance to the hop industry is hop stunt viroid. So this is the one that was discovered back in 2004 at a field in Toppenish and was basically the onus for us creating our own certified planting program in the National Clean Plant Center.
What is the effect of hop stunt viroid?
Well, the effects are basically in the name. You tend to get shortened internodes. The plant won't reach the wire. Yield will be reduced anywhere from, and some of the high alpha varieties it was negligible, but then you get a variety like Glacier, it would be as much as 65%, 67%, basically leading to destruction of the crop itself. The plants just eventually. The root mass becomes very constricted. They just become very droopy. The leaves become very brittle. Cones, they tend to, because one of the things in biology and plants in general is when they get stressed out, they tend to just go into reproductive mode. It's like, oh, we're dying. We have to produce babies. So they'll throw out a whole lot of really small, tiny cones that aren't really, to type or not really what you want in the brewing process as well, because the lupulin, of what gives us all the good oils and acids, the actual lupulin counts within these cones can be reduced anywhere from 60 to 80% as well, too. So aside from the morphological effects and the yield losses, you're also seeing influences on like the polyphenol, terpene content, oil contents, and these can all lead to a hop that might be genetically true to type, but phenotypically out of spec.
Interesting. And then what, when these things start taking hold, what's a typical kind of progression within a hop field? Something like, it seems like if this hits one plant, it's probably not the end of the world. But as you mentioned, these things, tend to grow and metastasize in some sort of, what does that seem like? And obviously you all are working to keep that from happening. So hopefully you don't see it that often, but when it does happen, what does it look like?
Well, in a commercial field in Citra, over the course of 4 years, the content of the parvoviruses, that's your hop mosaic and hot latent virus, went from basically 1% to 2% to 80 to 100% in 4 years. So again, these are aphid vectors.
4 years.
So
Wow.
This is why, again, like starting with clean material is very important, because over time you're going to get viruses. It's just inevitable with the nature of our crop. They spread very rapidly in a hop crop as opposed to some new woody species like tree fruits. And so it's just inevitable, and it's kind of an accepted part of the practice. And now mechanical transmission can be mitigated with certain farming practices. And in that same field over the course of 4 years, infection went from, the background infection of 1% to 2% up to about anywhere from 10 to 15%. So it's much slower. However, you can pretty much track the spread of the Eiler viruses and the viroids through a field by the management practices. So one really common practice in the hop industry that I understand why it's done, it's quick, it's efficient, and it's, keeps labor down, is mowing across the top of the hops. So when they're growing, like one of the early cruding methods that they do is just take a lawnmower and just, run it across the field. It gets the job done. However, the sap transmission that gets under those blades, you can almost trace a path of infection, from one row to the next. They tend to be very spatially confined when they're mechanically transmitted as opposed to insect vectored.
Right. So that early season pruning, when you want to cut down those first shoots, make sure that you're getting, putting more energy into the good ones that are to come. That whole process, blade to blade, it can then spread this throughout an area of the field.
Yeah, well, and there are
Fascinating.
sanitation methods that are
Terrifying.
very effective for cleaning your equipment in between fields. But again, it's a time and a labor constraint. And, while there's the perfect way to do things in the real world, there's also the realistic way. And farmers often have to balance what's best with what's realistic.
Sure. So we've been talking, throughout this about, that kind of mitigation. What you also mentioned, like cleaning up, the hops. When we're talking about hops, everything's going to be planted from a cutting. It's not going to be planted from a seed, because these all need to be female plants. And so, there is some concern there about making sure that the plants and the cuttings that you're planting from are also not bringing virus into a field.
Yeah, I think actually, Ryan, this might be a good one for you to speak up on.
Yeah, I can take this one. Yeah, like Megan mentioned earlier, there's some questions about seed transmissibility, but that's a great point for hops, that they are clonal. When you plant a hop seed, even if it is a female, it's not going to be resembling, even remotely, sometimes the mother that it came from. So we are relying on vegetative propagation or asexual propagation, whether that's from rhizomes or greenhouse cuttings, to propagate a citra or a cascade hop. The process for cleaning up a variety. So plants, when they get a virus, viruses infect plants the same way that they infect animals and humans. Spreads throughout the organism by its vascular system. So in plants, that's the xylem and the phloem, the water conducting, mineral conducting, and then the sugar conducting parts of the plant. But unlike animals, they don't have an innate immune system. They don't have any cells that are going to kill off and exclude that virus. So once a virus starts spreading throughout the plant, it's systemic and it's basically lifelong. There are conditions that can reduce the titer. So a very cold conditions, winter, and then very hot conditions as well can reduce the titer of those viruses, but they're still there. And they're still going to spread throughout the plant, especially for the viruses that Megan mentioned, the carloviruses and the iloviruses. One of the proteins, one of the genes that's encoded in those genomes is a movement protein. So that helps that, actually facilitates the movement throughout the plant. The exception to that is where there is not a vascular connection to a cell. And the primary locations there are the apical and root tip meristems. These are not yet connected to the phloem and the xylem and don't have a transmission route. There's no way for a virus or a viroid to get into those cells. So the way that viruses are cleaned up in any plant is really trying to maximize the size of that meristem and then excise it, graft it, or grow it in tissue culture. And with any luck and with good practice, you will have a clean plant because you've excluded those infected cells below that point. So as long as you're very clean and very delicate about it, that is the way that viruses are removed. And from that point on, once you have that initial cleaned, that single
Ryan.
clean explant. Yeah.
Ryan, I was just going to say, can you just say, like, how many shoot tips do you guys typically have to excise to get? It's very difficult to get a clean plant from meristem tissue. So how many explants do you guys have to try to make in order to get even one that might be virus-free?
Yeah. The success rate varies a lot based off of the pathogen, the season, the conditions. As I mentioned, the titer can be different, especially in very hot conditions. The success rate can be anywhere from about a 50% for something that's relatively easy to remove to almost impossible. So hop latent viroid in particular, it's extremely, extremely challenging to remove. And like the Clean Plant Center has done a lot of work into trying to figure out new methods to remove that viroid using cryotherapy, liquid nitrogen fracturing, and things like that to kill off some of the infected tissue and just get a really clean meristem that's not impacted by that treatment.
So then you have this very, this isolated, clean plant, and now you've got to grow it up into a quantity now that you can plant a field with, which, so that's the next phase of this whole project.
Right. That's a big challenge. Once you've got that one plant, you're really reliant on your processes, on just your logistics, your lab processes, your greenhouse processes to keep that clean. So going from one plant to millions, there's a lot of opportunity to reintroduce something. And once that's introduced, even if it's at 1% infection level, you've then just spread that throughout your entire propagation chain, and exposed any other varieties that you have in the greenhouse at that time to, say, the carloviruses that are aphid transmitted.
It's been pretty fun. I've watched when I was in Žatec in the Czech Republic, watching them take these plants under a hood, lab coats, lab environment, trying to then take these kinds of cuttings and build, and then grow them in medium to keep those plants healthy and virus free, and create all of these baby plants. It's quite a process there to go about doing that, and it's something that I think we all take for granted. You look at a giant field of hops, and every single one of there's a lot that goes into creating a field of healthy plants that's that big. Let's talk about how you all evaluate over the course of a growing season, kind of keep an eye on virus, check for that kind of virus. Are there visual tells? Do you do, a spot kind of testing? Obviously, we know, farmers in the fields get to know their fields pretty well, and they get to know those plants. And when they can see things happening, they've got early signs. And obviously you want to catch all of these things as early as you can. What are some of the ways that farmers look out for those kinds of things?
I think that can be tricky because visual cues in particular are oftentimes misleading. There are a lot of things that look like they would be pathogenic that aren't, whether that is there's temperature-induced stress, there's herbicide damage, all kinds of things that can present as virus-like symptoms that are not necessarily virus in origin.
Nutrient
and there are many others.
Edges of fields, wind, sun, extra sun, all of those things. Sure.
Right.
The edges of the field are already going to look the worst because they're the most exposed, and and that, of course, that's what we all see as we drive by them, right?
Exactly. And then Megan mentioned earlier too, these do not present the same way in every individual. There are, say, the apple mosaic that she mentioned earlier, that shows very classic ring spot, like kind of plant Lyme disease. You just have these bullseyes that are on the leaves or oak leaf patterns that are very visible in certain cultivars and then absolutely symptomless in leaves of others. The visual cues are, when you have a sensitive cultivar, it's a good indication, but it's not the most reliable way for finding those. So we're reliant on using molecular techniques most of the time, particularly for guaranteeing that the initial plants that we're propagating are fully clean. So we're mostly using quantitative real-time PCR. Just a PCR-based technique to determine whether or not it's there. And then there are some sequencing-based applications as well to see basically an untargeted approach, looking for all potential virus and virus-like pathogens.
Sure. Megan, same thing for you all. What techniques you all use?
Yeah, so it's a little different for us, just simply because we don't have a molecular lab for diagnostics on site, and testing every field can get very cost prohibitive for your average farmer. As Ryan said, like a lot of time, some varieties will show symptoms, some won't. There are, again, like most of the high alpha varieties that can be asymptomatic, but still contain virus and viroids in them that can still be spread elsewhere. And that's also something that we, take into account too. So, and with hop stunt viroid, you usually do get a latency period of about 3 to 5 years before you'll even see the physical symptoms. And so the viroid itself can be replicating within the plant and be spread before you even see the first, leaf mold or something in the enzymes. So just with that in mind, like I guess pretty much the rule of thumb is to start clean and do everything that you can to stay clean, with the understanding that over time viruses and viroids will accumulate. And as it's stated in that the economic paper, you do eventually hit a tipping point where if you know that you got a lot of a high viral load and a viral complex existing in your field, and your yields are starting to decline over time, part of that can be attributed to virus and to other things. And so usually the life of a hop field now, where they used to be around for 20 to 50 years, it's kind of rare to see one in the ground for more than like 7 to 10 years at this point. And that's not just a reflection of like wanting to start with clean material over and over. it's a reflection of the changing tastes and interests of the brewers and in the industry as well. So just the longevity of a hop field isn't as long as it used to be, and so that is a factor too. So we're just refreshing with clean plant material, and then just riding the field out as long as you can.
That's an interesting point. So where, when we talk about why hops cost more than they did 15 or 20 years ago, the lifespan of that hops field has come down dramatically because the expectation for flavor and aroma from brewers, what they expect out of that, when there was just alpha, and as you mentioned, alpha is much less affected by this than, say, some of those other compounds, you could let it roll longer and still get reasonable, acceptable alpha out of that same field. But with our increasing focus on aroma and flavor, it's just not acceptable, and brewers won't take those selections in the way that they might have if it was just going into alpha. That's really interesting to think about how, and that's a relatively recent phenomenon. This is only last 10 or 15 years that we've seen this kind of thing happen. Well, let's. I want to talk a little bit more about looking at building in more resistance in plants. I know that is a focus for various breeding programs to try to find specific plants that express resistance to all of these, various viruses and viroids. And then how you all are managing through that, and what that actually means in terms of the way that these plants are hitting now, and then some of the testing. I know, Ryan, you guys have done some data and analysis looking at your virus-free plants versus some of these older existing kinds of things, and have some interesting sensory data. And I'm going to talk about all of that. But first, breweries don't just need a point-of-sale system. They need a one-stop shop operating system. That's where Arrived comes in. Not only does Arrived offer a world-class POS built specifically for breweries, but they also provide Arrived brewery management, e-commerce, and more to help breweries scale and thrive. From taproom to distribution, Arrived gives breweries the tools to streamline operations and boost profits, all in one powerful platform. Visit Arrived, ARRYVED.com to learn more. Also, missed the Pro Brew booth and their incredible product animations at CBC 2025. You can now experience them all right now at probrew.com. Head over to probrew.com and see the cutting-edge technology that's revolutionizing beverage production, like the ProFill rotary can filler and seamer, and the Alchemator alcohol separation system in stunning high-definition animations. See how their advanced solutions can take your brewery to the next level. Go to probrew.com today and experience the latest technology in beverage production. ProBrew, brew your beer. Also, if you're launching a brewery or acquiring an existing one, consider our Brewery Workshop and New Brewery Accelerator, September 14th through 17th in Fort Collins, Colorado. Over 4 days, we engage in panel discussions, technical brewery tours, networking, and small working group sessions to help you better understand and prepare for the challenges of brewery operation. Tickets are on sale now. Visit breweryworkshop.com for more information and to secure your spot. Let's talk about what virus-free planting looks like today. If you've got now a 7 to 10 year field life, that means a lot of you all are replanting these fields a lot right now. And so there's a much more rapid turnover of these plantings. What does that turnover now look like? And talk to me about the nature of, what of these virus-free plantings, how you all are looking at breeding to then maybe increase resistance to some of these viruses within the plants themselves. Megan, why don't you take that one?
Well, I'll always take the planting out of a virus-free field. So typically what you do, like the actual starting from ground zero, is you have your plants in a repository at WSU at the Clean Plant Center Northwest. That's where the only place that's actually certified by APHIS to house what we consider Generation 1 clean plant material. So that is completely certified virus-free. You have to adhere to certain parameters, and the only facility that's able to be certified in that to that level is the WC facility. So we keep several lines in retention there, just again without having some of those lab resources to be able to do that in-house. And so when we want to start to plant a virus-free field of Azacca, for example, we'll go there and we'll get either they do rooted cuttings, they'll do virus-free rhizomes or a rooted flat of plugs too. So just depending on how fast we want to ramp up, we'll get that. And then we'll hand it off to a propagator, either in-house, or we contract with a couple of propagators too. And under certain clean plant propagating parameters, they'll basically, bulk it up to however many acres you want to plant. This could take anywhere from 2 to 5 months, really, just depending on how much you want to actually expand. So there has to be some foresight put into it. You can't just say in April, I want to plant 50 acres of virus-free Zoc in May. It doesn't work that quickly. So it takes a bit of crystal ball reading to kind of hope to predict what the market might want in some of these varieties the following year. So, but we'll again, we'll start clean, and then we'll just do everything that we can to try to stay clean over the course of the field. As far as breeding, that's definitely something I think Ryan would be better to speak on, because as of right now, in my program at least, we go in. I mean, we treat all our seedlings and our single hills as virus infected. They usually come out virus free, so that usually makes cleanup easier because there isn't a whole lot that's already accumulated in there. But actually, having breeding techniques, for viral resistance or viral tolerance is not something that I currently put in my program. That maybe, Ryan, that's something you can talk to you.
Yeah, so I guess from the breeding perspective, it's fairly similar at the HBC. So HBC is a joint venture between Yakutiya Franchise and John I. Haas. Each of us kind of have our own breeding teams that work on things collaboratively, and then we do joint releases together and they compete in the marketplace. Whenever you're adding in something like this, it kind of exponentially increases the cost to what you're doing. And it's very hard in my experience anyway, to run very controlled studies with viruses and other pathogens in particular, because if you're going to try to do it in the same microclimate in the same field, ideally, if you're planting virus-free material next to virus-infected, then that's going to change next year. So it's hard to control for all the variables well enough. That being said, we treat all of our breeding yards as kind of a survival of the fittest. So whatever is surviving well out there against all of Mother Nature's stresses, whether that is abiotic, temperature, drought stress, etc., or biotic stresses like powdery mildew, downy mildew, mites, and viruses, that is what is elevating at the top of the breeding program. And there's a certain amount of it that's been done since the early days of hop breeding in the United States. So, for instance, Megan mentioned Glacier a while back as being very particularly sensitive to hop stunt thyroid. Well, there's other cultivars out there like Nugget, which is hardly gets affected at all. And that is, whether intentionally or not, a result of living with this virus, living with these pressures, and finding those varieties that are surviving well against the stresses.
So just so I'm perfectly clear, what we are not then talking about here are virus-free plants that have been genetically modified or in some sort of way manipulated into having any kind of resistance to these things. You are simply doing it the old-fashioned way of, hey, this plant did really well. We should take a, cutting rhizome. How do we take this one that seems to be doing well? Check it, make sure that whatever we're taking from it is 100% virus-free when we start. But that there's not necessarily, when we talk about virus-free, we're not talking about something that is inbuilt in that plant's genetics. We're simply talking about the state when it's planted, and that it will very likely also at some point be exposed to that, and and then follow the same kind of impacts that we've now talked about.
That's exactly right. There's no genetic modification or anything like that is used in the removal process. It's all a physical removal, physical exclusion of that virus-infected zone. The breeding work that I've been talking about, that's all traditional breeding. There is, say, marker-assisted breeding or marker-assisted selection, or even using genetic tools to look at the effect of viruses and viroids on gene expression or epigenetics, but that's all happening at the research level, and just studying what is being affected by these pathogens, not trying to manipulate things to eliminate them or to mitigate their effects.
Megan, is there work, obviously you've worked, in that kind of university setting also, what is there anything on this kind of hops genetics side, trying to identify that kind of, virus resistance, at that kind of core genetic level?
I think more than anything, as Brian was saying, it's more, I don't know that there's a single hop out there that is actually resistant to being infected by a virus. It's whether or not it expresses the symptoms of said virus. So, again, like for whatever reason, the high alphas tend to be able to tolerate the viral load and infection quite a bit more. However, they're still carriers. So, and there is marker-assisted selection to breed against some of the other diseases like downy mildew and powdery mildew. However, those are still, and that they'll, if anything, they're just tools in the breeding process. But breeding in general is still heavily traditional. It's there's I'm not sure how brewers feel about genetically modified hops in their beer, but I know that there's growers in particular are resistant to the idea. And it's also, hops are so heterogeneous genetically that it's almost, there's so much variability that already exists within the genome in and of itself that even within one variety, as I know we'll talk a little bit later, we'll see drastic differences in terroir and just different characteristics due to stress or climatic conditions, things like that. So yeah, as far as breeding for it and selecting for it, there's it's really just observation.
Well, that frames the conversation in a more understandable way, so that this idea of virus-free is just a slice in time, for any given variety, any given field, from any given grower. Well, then, so then let's talk about that. If you're now getting 7 to 10 years out of a field, that changes the kind of yield dynamics that you're going to get from it. Obviously, we all know young plants are not going to necessarily, they're going to take a little bit of time to get up to full kind of production. Baby plants tend to, take a year in order to really get set and start producing at the level that people want it. And so a 7 to 10 year replanting schedule means you've got a lot more baby plants all of the time, all over the place. Maybe Ryan, walk through now what you all see within some of the varieties of the time dynamics, for these. You plant a new field with some fresh virus-free stock. You had a first year harvest, second year harvest. How long does it take to get into what you expect to be true to type for that hop variety coming out of that farm? And what are the general kind of dynamics from that now?
Yeah, that's partially location dependent. So in Yakima and southern Idaho, we're blessed with a long growing season and can expect a crop in the first year, especially if it gets planted early enough. And in hops, they're dependent on both photoperiod and length, number of nodes to flower. So if they are at the right number of nodes at say a little bit after midsummer, they will flower and bear a little bit later than a mature hop will, but they'll still bear that year. In Oregon and in some of the other cooler growing regions, there's not enough time between when you can get into the ground and work it and when harvest is. So typically plants are planted one year and then you start harvest next year. And even so, that's still kind of a baby crop. So it will take 2 to 3 years to get into full production, to hit that yield plateau that we'd like to see. And once it hits that year 3, then with virus free, we're trying to extend that plateau out as long as possible. But I think that there's some quality aspects there as well, that they really start fully expressing the full variety character when they are mature.
And when we say virus-free, this is just compared to, say, previous generations where they wouldn't, these cuttings or rhizomes that you would plant would have just been taken from, without this kind of attention to that being a virus.
Right, and that's partially where that plant size comes into play, because the way that we used to do things, even when I started back in 2019, was largely rhizome propagation. And well, you can go into a field as soon as the ground is workable, cut out rhizomes, and I need 80,000 rhizomes to plant my next field. I've got it, or my neighbor's got it, and I can go get those dug and planted, fairly quickly, even before it really starts to warm up. And those plants have a long time to really establish themselves and get a lot better yield potential that way than some of the especially greenhouse pots that are coming straight out of the greenhouse that are pretty freshly established. They're a little more fragile. They're not able to tolerate the wind stress, the sun stress, or UV and cold, as well as a rhizome planted hop that's coming out in situ would be able to. So there's a narrow window where we can actually plant those in the ground. So there's not as much time, and there's a lot more sensitivity around the planting timing, and then being able to get a good quality yield out of it at the end of the year.
Yeah, but that's another good point too. Ryan, since you bring up the rhizome propagation, is something that we'll do on our farm is when we do plant a clean field, we will use that farm as a root source for other. We'll use that field as a root source for other fields for up to 3 years after it's established. So again, like knowing that eventually it's going to become infected, we'll still consider it relatively clean in those first 3 years and use it as a source yard for other cleaning yards. Right, and then that also helps save on the cost too, because that is a significant factor. Is the cost per plant for a potted plant that's clean can be anywhere from like $1 to $2 more than one that's not certified.
Yikes. And so then let's talk about data there. When I hear brewers anecdotally talking about how say a virus-free plant didn't give them, or a field didn't give them the same aroma and flavor as whatever they were using before, it sounds like that may not be due to the fact of it being a virus-free plant. It may have other factors involved. You guys have some sensory data around this, Ryan?
We do. And I guess just to go to look at the public literature, there are documented differences, particularly out of some European publishing groups and research. There's some research to suggest in European cultivars in particular, there are changes to the essential oils. That being said, those changes are fairly minor in relation to microclimate and farm differences. So I think at the macro level, you don't see as much of that change as you do at the micro level. And that is where I think you see individual lots that maybe they are virus-free, but that's probably not why they're muted or they're not expressing as much. It's probably a little bit more to do with the specific lot level, whether that is the field age, location, or even the pick date. As over the course of the harvest window, oil is increasing over that entire time. And if you're picking up the early end, it's going to be expressing differently than at the end of the pick window.
I'm trying to wrap my head around why that would be different. This is still a part of the same kind of plant, just from a portion of the plant that doesn't have a vascular system to carry this virus into it. And so theoretically, the genetics are, exactly the same as, it might be otherwise. Explain to me how that could actually, produce a plant that might have some sort of, different performance in a way that is separate from all of those other environmental factors, sun, rain, nutrition, soil, growing conditions throughout a season. It seems like that it would be those other factors might be larger here. I'm not trying to, be an apologist for this whatsoever. I'm just trying to, as a liberal arts major, understand the science here and how that might impact some of these things.
Well, it's all context dependent and very complex. Viral pathogenesis affects many different areas of plant biology. It's, in particular pathogens where there's necrosis, you're affecting apoptotic cell pathways, and actually inducing cell death. There's changes to photosynthetic proteins and chlorophyll that leads to chlorosis. And then there's secondary metabolites, particularly like lignin biosynthesis, polyphenols that are involved in defense. And typically for like essential oils, they tend to be a little bit more correlated with insect herbivory. There's certain compounds like ketones that are associated with more plant defense against herbivores and against chewing insects in particular. But there are still likely some changes that are due to virus and viroid as well. Just changing the overall growth habit, opening up the canopy a little bit if there's not as much leaf material, and maybe opening up the cone structure too. So there are a lot of ways that there might be secondary impacts on essential oils, even if there's not a direct influence.
Megan, have you all seen any? You mentioned all the drawbacks of these viruses and viroids. Are there any even short-term, upsides in terms of, expression of, sensory expression in, flavor and aroma? Maybe even in some of these before, in this kind of like pre-discovery carrier type of phase for some of these viruses. Has anyone done any research around that we could, look at or think about, and maybe would there ever be any positive to that?
Well, one thing that is kind of interesting is virus-free plants, and one thing that we've always noticed, especially in a variety like Cascade, tend to be a lot more susceptible to other pathogens such as powdery mildew. And the reason for this is because when they're virus-free, the leaves tend to grow really lush. They're, soft, susceptible. And so, powdery mildew is a fungus, the most important disease in the American hop industry. Definitely causes most problems and requires the most intense management out of the, cadre of pests and diseases that we deal with. And we have a virus-free Cascade field that's going on 10 years now, so obviously not virus-free, but it tends to always, the best sensory, out yields all the other Cascade fields. And it's just I think it's a combination of just a really good location and, all the stars aligned to just make this field just be a bang for as long as a surprise. Now, one of the side effects or one of the symptoms of virus infection tends to be stunted, down-curled, waxy leaves. Now, what this does, unintentionally is basically makes those leaves hardened off from other infections like powdery mildew, for example. So while it does confer, I guess, a level of, kind of superfluous resistance, it's still the effect of the virus. It's always going to be a balancing game. So any beneficial trait you might get from virus infection is going to be outweighed by and potentially the bust you get as well.
But you all don't have data that says, the additional stress layered in from these viruses may produce, additional oil or beneficial oil within the plant. Because as you were saying earlier, Ryan, the primary driver of that additional oil production is, resistance to potential herbivores that might otherwise eat the plant and prohibit it from, doing what it normally does, which is, flower and make babies.
Yeah, and I think you mentioned, or you had said it well earlier with your talking about like the multivariate equation of what drives hop sensory and hop chemistry. Like hops, when they get viruses, it's just a kind of a steady load that is just a burden on the plants in the first place, and makes it less capable of dealing with other stressors. And I think that a lot of the time when we see differences in quality, it depends on when you get that stress and how intense that stress is. If you get it early in the season, it's probably not as impactful as if you are getting stress during gland filling and accumulation of alpha acids, essential oils, etc. So if you're experiencing water stress, drought stress, and heat stress at that time, absolutely, there's lots of research out there that suggests that those do have a major impact, and viruses are just decreasing the ability of plants to deal with that.
Judy, it's also, Ryan alluded to this earlier as well, it's also really hard to study the effects of just a viral or virate infection on hops and actually be able to attribute any type of phenotype that we get out of that to just that one infection. There's a grad student at WSU right now that my heart goes out to him. He's trying to research the individual effects of a few different viruses on a handful of different cultivars. And just being able to, first of all, successfully infect, first of all, you have to get a plant completely free of all viruses and viroids. That's a challenge, especially with the latent viroids that Ryan discussed earlier. And then you have to be able to successfully infect it with an isolate of a specific virus or viroid, and then maintain that singularly throughout the course of the study. So, as we said before, like hops just pick up viruses, through various different ways. And so it can be really challenging to actually just look at a plant with no other pests or diseases and just that one virus or virate and be able to actually say that this is the reason that we're seeing this trait. And so and that's why like they're, just doing literature review and preparing for this, just said, wow, there's still so many areas that need to be investigated. And it's just a challenging subject matter.
I think about when my kids were little going to, daycare or pre-K, and the number of diseases that those kids brought back and shared with us, and that's a class of 20, 25 kids. You got a hop field that's got, hundreds or 1,000 plants in it. You got all of these kids out there in the field together, in pretty close proximity with each other. And so it's not shocking that someone catches something and one of the plants catches something and it kind of moves through all of that, especially when the same things are touching a lot of the same plants all at the same time. That's a crazy metaphor for me to think about now as plant hop binds as children in a daycare. But
it's an apt analogy.
Yeah, I think so.
Just thinking about it, makes me shiver. Let's talk a little bit, again about the sensory on that, Ryan. You guys have been studying this, and you guys have some data. Share some of that, that data for some of the virus-free plants. And is there, when we talk about this data, I guess we probably even shouldn't talk about it just in a kind of a broad aggregate. We could even talk about it on a variety basis, because I imagine that certain hops varieties in this virus-free, cultivar approach may be a little different than others. If we've talked about it already, we're seeing that. Are there some differences there that you all see in the way that some of the virus-free fields work versus some of the, maybe more traditional, plantings, or, what does that data look like from your perspective now?
Yeah, absolutely. We essentially only look at those kind of variety, within variety differences. Not only because, Citra is not a Simcoe, is not a Cascade, but also because each of those varieties took their own path and at different times. There have been likely introductions of viruses kind of along the way since the very early hops growing here, like cluster, through some of those middle years with Cascade and into the newer varieties now, where you have a lot more, not only individual pathogens, but diversity within those pathogens that is out there. And so, when we're looking at, say, a Citra that might have been planted before we had a lot of the other isolates that are around for newer varieties. I think it's important to make sure that we're not confounding those variables. But for when we're looking at sensory, we try to look at it at the broad aggregate level, as well as at individual regional levels, and kind of focusing in on mature stand, because that is where you're getting into the maximum plateaued yield potential and the quality. At the aggregate macro level, there's, in our experience, not a lot of difference. There's not statistical difference between the two planting styles. Not to say that there isn't differences at the micro level. So at the individual lot level, individual field, individual grower, of course there's going to be differences. There's a range of expressions within each of these cultivars, and that's expected. But when you look at virus-free versus non-virus-free, there isn't that on that aggregate scale.
Have you all ever done any plantings of virus-free plants next to, say, a more traditional, kind of cutting, rhizome kind of planting within the same field, same terroir, or at least, close, with enough plants to try to, obviously you need more than one of each plant in order to have some sort of statistical significance and have it be meaningful in some sort of way. Otherwise, it's not science.
And I think that's definitely the challenge. Like, unless you're harvesting 30 plus plants out of each plot and testing those each individually, you lose the statistical power there. So we kind of rely on those broader scale comparisons, at least at the regional level, to make sure that what we're seeing quality-wise is not off-brand. That we are seeing that these are still true to type. They're every bit as much a Citra. A Citra is a Citra is a Citra, no matter if it's virus-free or not. and make sure that we're not seeing those quality differences at that macro level. But I mean, yeah, it's in an ideal world, of course, we'd love to see that at the individual field level. But yeah, it just gets challenging.
Sure. We're still working on brewer budgets here ultimately, and the plants can, those hops can only get so expensive, and you can only do so much research on it and still, hit the kinds of budget and price numbers that folks need it to be, so that we can give pints to drinkers at the same kind of price that they would like to pay for them. So
I do know the Cascade field I mentioned is probably the only one that like can actually say this with confidence in our field is next to another Cascade field that was pan of a rootstock, but right around the same time, and that'd be year or two difference. And the yield is statistically significant, like, within like 10 to 15% difference. Again, like the virus-free field tends to be one that, brewers tend to love a lot. I'm gonna make that's kind of anecdotal because brewer preference is about as highly varied as hop growth in general. So, but just from my visual observations, it is vis-a-vis it's at least in a variety like Cascade, which is pretty susceptible to some of the viruses, pretty obvious. But and then you got our virus-free field next to our virus-effective or rootstock source chart, I should say.
So we're talking about at worst a mixed story in in the way that some of these, and that in a lot of sense, again, pretty complex to try to pull these things apart, and that some of those differences, even if we attribute them to virus-free plants, may be attributable in other kinds of ways. And we have the kind of the summary, which is maybe one of the least satisfying, that there is so much more work to be done and so much more science and research that that everyone is engaged in, and that these are still questions that are being addressed at the research kind of level. And thankfully, we do have great research institutions. We do now. Certainly, some of that research is being impacted negatively by what's going on in the United States right now. We hope that we can kind of keep some of these important programs going, because they are deeply important to the kind of agricultural performance of plants in the fields and the growers, and you depend on. But I'm not going to soapbox on that for too long, other than to say that government-supported research is an economically important thing for the agricultural industries across the board in the United States, and we simply can't lose that and remain competitive and also efficient, and do the things that our growers need to do, our farmers need, in order to produce these ingredients that our brewers need. Let's just sum up with a kind of a forward-looking thing for both of you all. What are you working on, in this kind of virus-free realm over the next couple of years? What are some of the things that you're keeping an eye on, looking forward to testing more of? Things that either may give you pause or may get you pretty excited. What are some of those concerns, and what are some of those kind of motivating factors in this kind of realm? And Ryan, maybe we'll start with you on that.
Yeah, I think the biggest excitement, I suppose, is just looking forward to where we're at right now as an industry, where we're probably roughly around 25 to 30% of hop acres in the U.S. are the virus-free. And in the short term, we're looking at trying to implement that across all acres. It's really a kind of stewardship question for us, that we know that there is a major yield benefit. Like Megan said, with Cascade, even 10 years old, still seeing 10, 15% increased yields. Public research out there, depending on variety, you're looking at between 25 to 75% even in some varieties, like that Glacier example I mentioned earlier. We see kind of in that realm across varieties that we work with, and it's just leading to sustainability throughout the supply chain, whether that is at the grower level. You're able to get as many pounds on fewer acres, less inputs, less labor, trickling down into to the brewers, able to offer a lot better pricing and contracts, and just better for the planet too. Producing more hops on fewer acres of ground is just a good thing. So that is exciting to see the impact of the work that's been done at National Clean Plant Network, here at XG Franchise. And I think in the medium to longer term, just looking at the threats that you'd asked about, there are other pathogens out there that we know of. In Europe, in particular, the citrus bark crack thyroid, which is kind of like an Uber hop stunt. It'll outright kill crowns within a few years in sensitive cultivars, and that's definitely a major concern of ours and something we're monitoring for. Others,
and we something detected in Brazil.
Yep. It's in Brazil. We know it's in pistachios in California. So it's around. Could get in through, whether it's compost waste or imported plants, which the Clean Plant Center and APHIS are trying to avoid, but it's probably an inevitability. Then other pathogens like Erebus mosaic virus causes pretty severe stunting and symptoms on hops as well. It's not really endemic in the U.S. Or in the Pacific Northwest right now, but could be. Of course, other pathogens, worrying about like powdery mildew strains and things like that, which are, in the grand scheme of things, probably a little bit more likely. But it's still something to worry about and have to address, whether it's from, looking at rootstock sourcing and like what we were talking about earlier, or from a breeding perspective and trying to generate new powdery mildew resistant and giant mildew resistant hops. So that's kind of what I'm excited about, and then what keeps me up at night.
Megan, how about you?
Well, I mean, of course, I agree with everything that Ryan just said there. I guess one more take is that I'm just really appreciative that after we've just been like on an information campaign for the last 15, 20 years now, informing the industry about the importance of team planting material and how to avoid, spread of viruses, why it's important. And, I'm definitely hopeful that, the education efforts are being recognized. I'm seeing farmers really, do their very best to implement sanitation practices that make sense on their farm. And, just to use an example, like hop stent was discovered in Japan back in the 70s. And, based on like identification, replacing infected planting material and crops and sanitizing equipment, they were basically able to eradicate it. Now, granted, that's a very isolated hop growing region, and that would be very hard for us to do that, especially just given our size and everything like that. But the potential is there, and I think that the more farming efficiencies that we find, as that's happening, regardless of viruses and viruses, we just have to become more efficient at farming, just with labor constraints and just the general nature of the direction of the industry. So I think that it's just going to become a part of our DNA to think about these things. I think going forward, and just the awareness of it is a big part of the battle. Sure.
Well, I think that is a great place to bring this to a close. With G&D Chiller's 24/7 service and support, your brewery will never stop. Berkeley Yeast bioengineers ordinary strains to make them extraordinary. Old Orchard formulates custom blends featuring specialty ingredients. Indie Hops Lorien combines traditional elements of noble hops with an elegant twang of modernity. Discover how Steel Chill Cups can put your brand on every pour at steelchillcups.com. Arrived is your one-stop brewery operating system. Head over to probrew.com to see their cutting-edge technology that's revolutionizing beverage production, and the Brewery Workshop will help you prepare for all the challenges of brewery operation. If you've enjoyed this episode, go to beerandbrewing.com, click on that subscribe button. We would appreciate you supporting our mission to bring you the best in brewing information, technique, creative inspiration, and everything else from not only the country and the world's best brewers, but also our smartest scientists like Ryan and Megan here. Got some good stuff on the horizon here with the podcast. Heading up to Vermont and Montreal next week. Going to get some conversations in with brewers up there. June, heading to Paris and Munich. Going to see what I can scare up for some conversations around those. Looking forward to visiting some brewers in those areas. Megan, if people want to learn more about Latitude 46, where can they find out more information about your program and the hops that you're working on there through Latitude 46?
You can learn more about us through latitude46.com, our website, and we also have Instagram handle, latitude46talks.
Ryan, if people want to learn more about the work that you're doing, where do they find more about you?
Yeah, we are at achievranches.com. We've got content on YouTube, on Instagram, and anybody that's visiting the Valley are always welcome to drop in.
Awesome. Well, thank you all so much for talking with me about virus-free hops. Ryan, Megan, cheers.
Thank you, Jamie. Cheers.
Cheers. Thank you very much.
This podcast has been brought to you by Craft Beer and Brewing Magazine for those who love to make and drink great beer. To learn more or to subscribe, visit beerandbrewing.com or find us on social media at Craft Beer Brew.